Good morning, afternoon, and evening to everyone joining us, and welcome to today's webinar. Today's talk is entitled Challenges, Regulatory Considerations, and Innovative Solutions for Designing Trials for Rare Diseases. My name is Alicia Gruszka. I'm a science communicator and medical writer working with TransPerfect and Springer Health Plus, Italy, and it is my pleasure to be your moderator for today's session. Today's event will run for approximately 60 minutes, including time for a Q&A session with our speakers. This session is designed to be interactive and works best when you're involved, so please feel free to submit questions and comments for our speakers by using the questions tool in your control panel, and we'll answer your questions during the Q&A session. If you require assistance, please contact me at any time by sending me a message using the chat in your control panel. At this time, all participants are in listen-only mode. Please note, this session will be recorded and made available to you on the IDDI's website for viewing within 24 hours after the webinar has ended. This webinar is proudly hosted by the International Drug Development Institute, or IDDI. IDDI is the Center of Excellence in Regulatory Statistics and Clinical Data Science for global pharmaceutical, biotechnology, and medical device diagnostic companies with a focus on oncology and ophthalmology, central nervous system, and rare diseases. IDDI addresses your intricate challenges, enhances efficiency, and minimizes risks throughout the entire clinical trial process. Founded in 1991, IDDI is headquartered near Brussels in Belgium, with a US Center of Operations in Raleigh, North Carolina. Now it is my pleasure to introduce you to IDDI's Clinical Development Strategy team and today's speakers. Dr. Elizabeth Els-Coart, PhD, is the Director of Statistical Consulting and Research at IDDI. Els directs the development, modification, and evaluation of processes for providing consultancy and study design support for clinical trials at IDDI. Professor Tomasz Burzykowski, PhD, is the Vice President of Research at IDDI. Tomasz is a professor of biostatistics, bioinformatics at the Data Science Institute of Hasselt University since 2002. In 2023-24, he served as the president of the International Society for Clinical Biostatistics. Okay, I'm going to change. Tomasz, the floor is yours.
Okay, thank you, Alicia. Somehow I do not see the presenters panel. Can you give me the presentation rights? Okay. All right, I hope that you can see my screen now with an outline of our...
Yes.
Hello? Can you see my
Yes,
screen?
fine.
Okay,
Yep.
fine. All right, sorry about that, but it's usually a bit more complicated with online presentations. So this is an outline of our webinar. So we'll start with a, let's say, 2 slides introduction to rare diseases to set the scene. And then Els will talk about regulations and challenges related to clinical trials for rare diseases and orphan drugs. And then I will try to look a little bit more closely on possible trial designs and statistical methodology with a couple of examples. So, okay. So here's some information on rare diseases. Now, there's no single definition of a rare disease because it varies depending on the country and continent. For instance, in USA, is defined as a disease which affects less than 200,000 people, so 1 in 1,700 individuals. In Japan, it is defined as a disease which affects less than 50,000, so 1 in about 2,500 people. And European Union and World Health Organization defines rare disease as a disease which affects 1 in 2,000 individuals. It's a large number of diseases. Orphanet, the organization, the platform for rare diseases, gives the count to about 6,000, 7,000 rare diseases. WHO indicates more than 7,000 diseases, which apparently impact more than 300 million people worldwide. 70 to 80% of these diseases are genetic and chronic. And most of them are pediatric. 70% of our diseases population are children, which is obviously one of the challenges which have to be taken into account when developing drugs. Unfortunately, more than 95% lack effective treatment. So that's obviously a challenge from the point of view of public health. As I said, rare diseases are defined based on the prevalence. There are also additional definitions, like there's the definition of there's no formal definition of an ultra rare disease, but for instance, in the USA and National Institutes of Health, they use a definition of an ultra rare disease as one which affects 1 in like 50,000 people. And that's actually a large portion of a fraction of rare diseases. So these ultra rare diseases are there, which means that there's not many people really worldwide with or across a continent with the disease, which is obviously a challenge in terms of the, for instance, availability of patients for clinical trials. There's also this paper by Smith et al. From 2022, which also coined the term of hyper-rare diseases, which is really like 1 in 10 to the power of 8, affecting 1 in 10 to the power of 8 individuals. So it means that there's like a handful of people all over the world with this hyper-rare disease. And they claim that actually these hyper-rare diseases are estimated to outnumber the other categories by orders of magnitude. In this webinar, we will not focus on hyper-rare diseases, but we will look at rare diseases and probably perhaps with examples a little bit more on ultra-rare diseases, so diseases where sample size is really a challenge. So that's more or less the scene, and I will now ask Els to take over and look into the regulatory guidelines and challenges of these trials. Let me try to change presenter to Elizabeth. Els?
Yes, I can see it.
Yes, we can. All right, I'll shut it up.
Yeah, you see my screen all right?
Yes, it's okay.
Okay. So let me continue with introducing the regulatory landscape for the development of orphan drugs. So the orphan drugs, the drugs that were considered abandoned by the industry due to little prospect of return on investment. And it was exactly this that the Orphan Drug Act in 1983 tried to address, where they were granting economic and regulatory incentives for developers of orphan drugs. And this has really been a landmark legislation, as we see that if we say about 10 drugs were approved in the decade prior to the Orphan Drug Act, you can see that in the decades afterwards, here shown in orange, that the number of orphan drugs that got approval has clearly increased. Also, here in blue, it's the number of orphan drug designations has steeply increased. And a designation, that's the specific status of a drug developer can apply for to have access to those benefits for rare diseases. So it means there's a lot of development, a lot of research ongoing on drugs for rare diseases. Here you can see that not all the rare diseases are treated equally, say. So if we look at all the designated diseases, and that is what FDA calls a rare disease that has at least one orphan drug designation, you see that the majority here as like 1 or 2 to 3 designations. And at the other end of the spectrum, you have diseases that have more than 20 designations. So a lot of research is ongoing and a lot of effort is done for just a handful of diseases, and they get really more than 1/3 of the overall orphan drug approvals in the US. If we then look at, like, since the Orphan Drug Act came into place, what the therapeutic areas are that got the most orphan drug designations and also approvals, it's clearly that in the space of oncology, we have more than 1/3 of both designations and approvals. And then you see followed by neurology, infectious diseases, and so forth. Of course, also by now, in all other regions, there are orphan drug regulations. In the EU, we have the EMA endorsing a regulation starting in 2000 with many designations, a lot of research ongoing, and many orphan medicines already authorized. Now, what all those orphan drug regulations have in common is that they encourage the drug developers to request this orphan drug designation early on in the development so that they can benefit from all the incentives that are available, the regulatory assistance, the monetary incentives, and also then after approval, the marketing exclusivities. What they also have in common is that they all acknowledge that it's so much more challenging to develop drugs in rare diseases and to perform clinical research for rare diseases. And those challenges, they are really, there are so many. And I will not go into detail on all, I will just mention a few. So if you think about the challenges for designing the clinical trials, of course, the small population size is one of the main challenges. Often there's not an accurate diagnosis yet, and that's often linked to the heterogeneity of those rare diseases. And then also, often there's a lack of consensus on the clinically meaningful endpoints, so there are no endpoints available that are like readily available to start and design your clinical trial. Also, once your clinical trial is running, there are also many challenges in the conduct, and one is related to the patients that might have difficulties in reaching the centers that we only, let's say, the very specialized centers will be involved in the trials. And you see that many of the clinical trials for rare diseases, they end prematurely because they cannot enroll sufficient number of patients. Once everything is done and all the data are collected, then those orphan drug trials, they are often more challenging in terms of analysis, again, because of the heterogeneity in the patient populations that might increase the risk of confounding. And also the type of trials that are used for orphan drugs are often more complex. So there's more, we need to pay more attention to the analysis approach to make sure that the treatment effects are estimated in an unbiased way. Now, although all those orphan drug regulations, when you read through them, you scroll through them, you see they all acknowledge the difficulties that are there. But at the same time, and I'm showing you here some text examples from the EMA and the Orphan Drug Act in the US, that they say the orphan drugs, they have to adhere to the same safety and efficacy standard that applies to all medicines. So in essence, the orphan drugs, they follow the general guidance for drug development, although that many of the agencies have, in addition to that, they have many rare disease specific guidance documents and programs. And here an example of some specific guidance documents that FDA has issued for rare diseases. It's not an exhaustive list, but you can see it's already a lot related to natural history studies, related to pediatric studies, and so forth. And FDA also has these interesting pilot programs, and those are not all specifically targeted to rare diseases. Some are, like the first three you can see here. And these pilot programs, they are designed to test some new approaches to regulatory support and to streamline the development for rare diseases. So for instance, the first one here focuses on endpoint advancement, and it means that if you access into this program, you have more access to the FDA advice on how to develop and validate new clinical endpoints, what is an important aspect in clinical research for rare diseases. Now, other programs like the one I mentioned here, the complex innovative trial design, that's not specifically targeted to rare diseases, but when Thomas will give some of the examples, you'll see that most of the participants in these programs, these are all developers of orphan drugs, and that's as expected, right? Because it is in this setting that often the trialists, they try to use novel trial designs, and they then seek some advice from FDA to understand whether this could or could not be acceptable. Yes, I've told you that the orphan drugs, they are supposed to follow the same efficacy and safety standards that are out there for all drugs. But it's also clear when you look at clinical trials for rare diseases in practice that it's really this standard is applied with a lot of flexibility, and that across all the stages of development. For us as statisticians, I think this is good news because it means that we can extend our what I call regulatory statistics toolbox. There are more statistical methodologies that we know of, but that we can use because we know that the agencies will be more open to accept those. And so to show it in maybe with a more, it's an elaborated graph, but it means that we all know that we need a certain level of evidence, a totality of evidence that's needed for a drug to get approved. And for rare diseases, there are many pathways that could generate this evidence. So it could include natural history data. It could be setting up natural history studies if these are not known. We could include PK/PD evidence from the dose optimization. We could think about augmenting the controls in the randomized trial. It could be a single arm trial. It could be an adaptive trial. Just to show you that there are many options or many different ways one can generate all the evidence that is needed, and the best way will depend really on the disease and all the characteristics thereof. Now, making things a bit more concrete. So we are dealing with small sample sizes, and we know that decision making needs a sufficient amount of data, high quality data. Now, what are our options? We could get more data or higher quality data without increasing the sample size. We could get more information from the same data, or we could consider that we might want to change the level of information that is required to approve a drug. Thinking about increasing the data available from inside the trial, we can do obvious things like not dichotomizing continuous endpoints, collect more baseline covariate data, which I think is very useful here for this heterogeneity in the disease. It might help us to further to avoid some potential confounding when it comes to the analysis. What's also important is that if at all possible, we can collect longitudinal data, right? So we have more data pertaining to one patient. And what I think is also something that, although it's very time consuming, we might improve the data quality by using more efficient endpoints. And I came across this example of the 6-minute walk test. And I think for everyone who has been involved in clinical trials, you'll have heard of this 6-minute walk test. It is a functional capacity test, and that's why it's so much, let's say, liked by clinicians and patients, because it is a functional test. It means something to the patient. Actually, what the test measures, it's the distance that a patient can walk in 6 minutes. So this is measured at the beginning of the trial, and then it's measured again after the treatment to see if there was an improvement. Now, the issue with this test is that it's tested in a clinical setting, so it's tested on a specific moment. And this might not be, let's say, the best moment for the patient. The patient might be tired, might be influenced by this test environment. So the endpoint is notoriously variable. And then they thinking about the type of information that was like really needed in this setting, and it's the primary endpoint in the Duchenne muscular dystrophy disease. People were thinking about how can we collect the same type of information but with higher quality. And what was developed is a new endpoint, a digital endpoint, that's called stride velocity 95th centile. And it is actually this, it's measured with a wearable that the patient can wear most of the time. And the endpoint is the 95th percentile of the stride velocity during the patient's daily life activities. So it's measured continuously on longer term. So it means it's way less variable. And that in turn means that we can capture this clinically meaningful information, but in a more sensitive way, so that the trials can be smaller and shorter, right? So it's a novel endpoint that was developed, but it is, I have to say, when you look at the timelines, it takes a long time before the endpoint is developed and before you get the regulatory buy-in. So here for this specific endpoint, the stride velocity, you see that back in 2018, it was granted qualification advice by EMA, so it was allowed to be used as secondary endpoint. In 23, after some years of experience with the endpoint, it was allowed as primary endpoint by the EMA. So it means that there are now clinical trials running with the stride velocity as the primary endpoint for the EMA, but for the FDA, where the endpoint is still under review, it is a secondary endpoint. But here for FDA, the endpoint is now being reviewed through this rare disease endpoint, acceleration or advancement pilot program. And so there is hope that soon it will also be acceptable as primary endpoint there, and hence increasing the data quality collected in the trial. Now, thinking further about increasing the data that's available from a small trial, we can also think about, can't we increase it with data that come from outside of the trial and borrow information from them? We heard a lot about the use of external controls, and that can be like to completely replace the internal control of a randomized trial, or it can be used to augment the internal control so that you can change the randomization ratio and have more patients offer the option to already receive the experimental treatment. Typically for rare diseases, the natural history data can be used, and all is in the with the idea of borrowing this information. Now, Tomasz will go further into detail on this topic and will also show that this is not without risk, and that here we have to really be careful as trialists and as statisticians that we can make sure that the treatment effect we estimate that it will remain unbiased. The other option we have is to maximize information that becomes available from limited data. And here we have to, I think it's of utmost importance that we as statisticians, we try to find the most efficient design and analysis for this specific indication. Of course, we always try to find the most efficient designs, but I think in this setting, it's worth some additional attention. And this could be what we call by now a simple group sequential design, a very efficient type of design that's out there for decades, that will introduce some interim analyses, futility interims that you can stop the trial if there's a low probability that the drug will be sufficiently active and the patients can move on to potentially other trials, or an interim for efficacy so that in case the treatment effect turns out to be more extreme, better than you anticipated, the drug can be approved and can be made available to all earlier. We can think about adaptive designs like seamless phase 2-3 trials, where we, in the phase 2 portion, we do a dose selection drop an arm, continue with one dose, but still in the end, for this dose, we use all the data collected within the trial for the final analysis. What I also think is important here to maximize information is that wherever possible, we use within subject designs and analysis. Like the endpoints that I was just mentioning, this 6-minute walk test or the stride velocity endpoint, this is measured at baseline and again at the end of the trial or after a certain period of time. And so you're looking at a change within this patient. What did the treatment do for this patient? And this within subject outcomes, they are known to be often less variable and also reducing confounding that might be there when you compare between subject measurements. Now, here we also have to think that randomization can play an important role. We have to think about randomization beyond the parallel groups that we are used to in for the drugs for the more common diseases. We can think about, whenever possible, about a crossover. We can think about randomized delay start, randomized withdrawal. We can think about methodologies that allow a patient to become his or her own control. And that is so powerful. Now, the last thing that we could potentially change is that if the level of information that would be required for a drug approval, if we could somehow lower that a bit, that could also help us. But that's, of course, that's up to the regulatory agencies to set the bar there. And it's not easy to see going through the guidance documents what is possible and what is not. You only find very vague terminology, or it's not like the official US let's say message. But if you go through some materials, you see that often it's mentioned that less stringent alpha levels may be used for ultra rare diseases. And this is also something that you can see in some of the papers, that when the disease is really rare, like not just rare, but ultra rare, then there is some margin here. So I think with this, I've set the scene of like, what are our options? What are the challenges? And I think, Tomasz, I can move it over to you to now go a bit deeper and show us some further examples.
Okay, thank you. All right, I hope that you can see my screen now. So yes, I will try to look at some examples from literature and from experience regarding trial designs and methodologies, statistical methodology, which could be applied in clinical trials in design and analysis of clinical trials in rare diseases. So Els already mentioned a couple of these, and she mentioned the use of external controls, and especially in the case of, for instance, ultra-rare diseases, it may be really difficult to convince researchers to have a randomized trial with two groups, a control group and the experimental group. So the idea of external controls come into the picture. And as Els said, you have to be careful about that because, yeah, there may be issues related to potential confounding. So this is an example of a review of related to a drug for pump disease, the drug called Myozyme, where actually a trial with 18 patients was conducted, a single arms the study, and there was a historical control group, and this is in the red rectangle you have like a snapshot of the conclusion. So the trouble which was found by the reviewer of the dossier was that the subjects in the study did not enter the study at the time of birth, so they had to survive long enough to enter the study. On the other hand, the historical control group was actually including all subjects, including those who died within the first months of life. So you had kind of a guaranteed time bias in favor of the study cohort. And for that reason, in this situation, the use of this historical control group was questioned. So it is important to pay attention, and there are multiple issues which might be encountered when selecting the external controls, because I mean, especially if you have small sample sizes, right? Even checking the distribution of some factors, clinical factors between the, let's say, the study group and the external controls may not be very helpful, because with a small sample size, there may be differences which will pop up there just because the number of patients in both groups will be small. So it is a challenge, but as I said, in an ultra rare situation especially, yeah, this idea of using external controls and just having a single cohort of patients subjected to a treatment is attractive and still entertained. Now, let us look at adaptive designs. Els already mentioned this this program with complex innovative designs. So here's a paper published in 2014. So it's a bit of, you know, like a decade ago, but it's a snapshot of what was going on there. So in this paper, the authors looked at, during this period of about 3 years, they looked at the 59 scientific advice letters which address questions on an adaptive design for a phase 2 or phase 3 trial, okay? 92% of these cases, of these 59 cases, these were trials which were planned as confirmatory and labeled as phase 3 or phase 2/3 studies. Interestingly, majority of them, like 80% of them, were accepted or conditionally accepted. Now, what is interesting is that about 60% of these cases were related to trials for a rare disease. And in fact, 44, so out of 59, so 75% of these cases proposed a trial as the only pivotal study. Among these 44 single pivotal study cases, 68%, 30 were on rare diseases. And moreover, out of these 30, about half had the orphan designation. So what I'm trying to tell here is that these adaptive designs, as Els already mentioned, were really very much linked to trials in a rare disease situation, and they were actually used as a kind of a way to overcome the challenges of a trial in a rare disease, which Els already mentioned. So here's an example, not very concrete. This is an example reported in this paper in Trials in 2014. So this is one of the examples of the cases considered in this scientific advice letters. So there was an open label to armed single pivotal phase 3 study to show superiority of the drug over a standard treatment for the primary endpoint on overall survival. So this was an anti-cancer drug. So it was like it was oncological destination. And the trial was designed with 2 interim analyses at 50% and 80% of events because the endpoint was overall survival. And the idea was that at the second interim, there could be a sample size increase. So actually not really sample size increase, but event count increase by extending the follow-up of the accrued patients. High-beta Peter boundaries for efficacy were used to design the trial, and inverse normal combination of test statistics was supposed to be applied in the analysis. Now, the advice of the scientific advice working party was that this design was acceptable if the type 1 error rate was controlled and operational bias was avoided. However, they indicated that at the first interim analysis, stopping only for futility could be considered. So at this early analysis, at 50% of the patients, they would not like to see stopping for efficacy. Also, they questioned the use of the inverse normal combination of the statistics for the analysis, and they actually advocated the use of the standard fixed sample test statistic. But this example shows the thinking of, well, let's say researchers into direction of the adaptive design, and the advice obtained from a regulatory agency was that indeed this type of design might be acceptable under some conditions which were stipulated as I have just explained. So adaptive designs are a possibility, especially perhaps, for instance, in the context of, let's say, rare variants of oncological diseases. Now, let us look at some at the general approach, which is very often entertained, that is Bayesian design. So in rare diseases, Bayesian approaches are, in a sense, quite natural, okay? Because as El said, with small sample sizes, you would like to enrich the information which you have in your data. And small sample sizes associated with rare diseases benefit from incorporating auxiliary information to augment statistical power, but that's a natural step towards the Bayesian framework. Also, as I mentioned myself, most rare diseases are pediatric. They are most patients are children, and then borrowing information from adult studies and in designing trials for pediatric population for a rare disease also is naturally achieved by using Bayesian framework. Now, this is this complex innovative trial design pilot meeting program. This is the FDA program which Els mentioned, and the idea is that sponsors proposing an innovative design may engage with the regulatory staff at 2 meetings where they get advice. So in 2021, 100% of 5 selected designs, so there were 5 selected designs, and all of them were Bayesian. These are the diseases, and Duchenne muscular dystrophy and pediatric multiple sclerosis. These are clearly 2 rare diseases. So we can see that, you know, out of these 5 designs, which were actually Bayesian and which were selected in this program, already rare diseases were present. Now, this idea of borrowing historical historical data via Bayesian approach is quite natural, as I said. You can actually use 2 versions of borrowing, where with a static weighting, so with fixed weights which are assigned to the historical data, or with dynamic weighting, where weights depend on the agreement of, let's say, current data with the historical data, right? So this is like borrowing from trials which have been already run. So usually we talk about 2-step approach. So first we construct historical data prior. So D0 is the let's say historical data. So we have like a prior for our, let's say, parameters in vector theta, and then we have likelihood, which is actually constructed conditioning on the historical data. And then this historical data prior is then combined with the data in our study to come up with the posterior, which can be used for inference about the drug which is considered investigated in the new study. So usually this prior, which we use to construct historical data prior, is vague or even flat. And very often so-called robust mixture priors concept is used to construct these priors. I will come back to this idea later, but that's that general idea that if we have historical data from trials, we can actually construct a prior and then use this prior to actually analyze or design our trial. This can be used, this idea of borrowing from historical data can be used to inform design, as I said, for instance, to provide priors to simulate scenarios. It can also support internal no-go-go decisions. For instance, in this case, we may want, a company may want to essentially use priors, but internal priors, which are let's say, expressing opinions within the company about the drug, and then use this Bayesian approach to sort of like influence the decision whether to continue development or not. Obviously, you could consider using the idea in regulatory applications. It may be problematic, but it depends on the situation. For ultra-rare diseases, maybe it might be acceptable. And well, remark is that, well, for regulatory agencies, for instance, the static borrowing sometimes may be preferred because this fixes the influence of the historical data on the results of the new study. Now, important idea which can be entertained both within the Bayesian framework but also outside it is borrowing from natural history. Knowledge of natural history is important. So we might want to have historical data on the persons we hope to enroll in our trial. And these data might inform us about like the priors, which we might want to use to Bayesian framework, but also it could tell us something about like what could we expect from patients in this disease if they are, for instance, left untreated. And that could be used also to entertain this idea of a crossover study, which Els mentioned, right? When each patient acts as his own control or her own control. So the idea would be that we could measure or evaluate the data at baseline, at the entrance to the study, and then the patient is subject to the treatment, and then the baseline data are used to sort of like quantify the treatment effect and evaluate the outcome of treatment. This idea of let's say, using the historical data or natural history and making patient his own control it can be seen as like more ethical approach to rare diseases in clinical trials, because it's not fully crossover. You don't come back from the active treatment to no treatment again, right? So you don't have this step there. But obviously, this idea of using patient as his or her own control, this might be seen as providing lower evidentiary value than a traditional two-arm randomized clinical trial. But again, as we said, randomization, especially in ultra-rare situation, may be encountered with issues. And then there may be also an idea of borrowing from PK/PD data. So along the process of evaluation of the drug, we'll start from the drug formulation, but then often early in the development, PK profile and PD effect might be measured, and then a disease and population model may be created, and then clinical efficacy and safety data will be collected. So in fact, in each of these steps, each of these steps can inform the next one, or each step can be informed by the preceding ones. And this is, again, a natural thing to do in a Bayesian framework, where each step generates a posterior, which can be used as a prior for the next step. So for orphan drugs, sample sizes are small, so PK data are valuable. And one could, again, as we said, a lot of rare diseases are related to pediatric patients. So applying PK considerations for pediatric drugs can be of interest here. So let me look at a couple of examples. So this is an example taken from this reference from the chapter 12 in the book by Lesafre, Bio, and Boulanger, in a book edited by these three researchers. So this is an example of progressive supranuclear palsy. It's a neurodegenerative disorder with the prevalence of 1 in about 17,000 individuals. And there were 2 randomized placebo-controlled trials, neither of which was successful. So as the primary endpoint in these trials, the PSP rating scale, a quantitative measure of disability, was used. So it ranged between 0 to 100. and it actually was constructed based on 28 items, which captured like 6 domains: daily activities, behavior, and so on. Patients were followed for 52 weeks, and the progressive supranuclear palsy severity was tracked. So for the purpose of coming up with a prior for a Bayesian approach, these placebo arms in these trials were pooled, and change from baseline in this score at 52 weeks was taken as the outcome. So the mean of the outcome in this in the combined 144 placebo patients was about 11. Standard deviation was about 10. And this is the idea of this robust mixture prior. So 50-50 mixture of this prior based on, let's say, assuming normality for these data, and a vague prior. So prior centered at the same mean, but with a very large variance, was constructed. And the idea of this robust mixture is that in a sense, it's a heavy-tailed version of the historical prior, because the tails. So here you have the illustration. So you have the red one is the vague prior, the flat one, and the green green dashed line is the historical prior. So this normal distribution, and then the mixture is the black line. And you can see that the tails of the black line are, let's say, coinciding with the tails of the vague prior, but the informative part of this mixture is coinciding with the historical prior. And this allows actually for cautious and data-driven borrowing, because if the placebo response in a new trial is close to the previous data, then historical information will be borrowed from this prior. But if not, if the placebo response is actually consistent with the previous information, then there will be little or no borrowing, because then we will be using the tails of this prior, and there's little borrowing. So this can be seen as a again static way of weighting. This can be also made dynamic, where like a hyper prior may be put on these weights. But this is the idea of sort of like using previous trials to come up with a meaningful prior for a trial. Another example, borrowing from historical data, not necessarily for the design, but for analysis, is Still's disease. This is a rare inflammatory multisystem disorder with a low prevalence, and there's a pediatric variant and an adult variant. And in fact, a drug, canakinumab, was accepted for the pediatric variant in multiple clinical trials. But then there was a question whether this drug could be also used for the adult variant. So there was a small trial, a small phase 2 trial, randomized, placebo-controlled, which was run in this adult onset variant, okay? However, it was terminated prematurely, with 36 out of the planned 68 patients included. And the primary outcome was the reduction of disease activity score at week 12. For that primary outcome, the difference in the proportion of responders was not statistically significant. So there was a trend favoring the active drug, but the trial was prematurely stopped. There was not enough, not large sample size. And then the amount of information about the drug for the adult variant was not large enough. So the idea was to use Bayesian inference to support similar efficacy for canakinumab in adult variant, borrowing data from the juvenile variant. And in fact, these Bayesian analyses contribute towards the regulatory approval in the US of canakinumab for this adult variant in 2020. So you can read about this example in this reference by Dunger, Baldauf et al. Published in 2022. So a little bit more about what happened there is that the Bayesian analysis of the adult study was conducted, borrowing information from the juvenile studies. So there were several endpoints which they considered. They used this idea of a robust meta-analytic prior, and then they used the posterior for each endpoint, which was based on the juvenile studies, and then also joining the adult study data. And for similarity of the outcomes, they use the thinking that the posterior median for the adult study should lie within the 95% predicted credible interval, which would be based on the prediction from the juvenile studies, from the prior based on the juvenile studies. So here you have a picture from this paper. You see these are 4 different endpoints, and in the red rectangles you have the predicted credible interval based on the juvenile studies, and then you have the posterior interval for the median for the adult study. And you see that in all the cases, the median falls within the predicted for the predicted credible interval based on the juvenile studies. In fact, the posterior credible interval for the median is actually included entirely in this interval based on the juvenile studies. Also, they looked at the PK data. So they looked at the exposure, and they split the information from the juvenile studies in different age categories, and they have concluded that in fact for all the age categories the exposure in the juvenile studies was comparable, and it was also comparable to the exposure which they actually observed in the adult study. So in terms of the exposure to the drug, the exposure was similar. And then they went to the PD data, and they looked at the association between the endpoints and the predicted concentrations of the drug, both again in the juvenile studies and the adult onset study. And there you could see that they saw that essentially the same association could be observed. So by using this Bayesian analysis and by borrowing information from the juvenile studies and from the small study in adults, this Bayesian analysis allowed to conclude that the effects of the treatment in the adult variant of the disease was comparable to the results in the juvenile study. And finally, an example of a disease of the trial where natural history was used. This is an example in Crigler-Najjar syndrome, again, an ultra-rare genetic disorder characterized by the life-threatening accumulation of bilirubin in the blood. So this is a genetically, this is a disease where genetic mutation prevents production of the enzyme which actually facilitates expression of bilirubin. So if left untreated, children with the highest levels of bilirubin may develop encephalopathy, so which leads to severe neurological consequences. So the current therapy is the phototherapy, daily phototherapy, and it partially controls the jaundice, but yeah, it's a very inconvenient treatment, right? So withdrawal of phototherapy should lead to increase in bilirubin level, and this means that well, patient could act as his or her own control, because upon withdrawal of phototherapy, you should observe the bilirubin increase. So then you could try it at the baseline and then see whether subjecting patient to the treatment and withdrawing the treatment would whether the withdrawal of phototherapy will without the after the genetic therapy, whether the bilirubin level would go up. Now, this for ethical and clinical reasons was not done. So instead, the researchers didn't check the increase of the bilirubin at baseline, but they assumed that the probability of the bilirubin not increasing would be very, very low. And the idea is that a gene therapy, which would sort of like re-establish the production of the enzyme, could actually help the patients. So in fact, a snapshot of patients from this trial was published in the New England Journal of Medicine. And they also, in the paper, you can have a supplementary information with the protocol. And this is what I said, is that, okay, they looked at as the endpoint, they look at the successful withdrawal of phototherapy, so not leading to an increase of serum total bilirubin over a threshold of 300 micromole per liter. And they assumed that the probability of such a successful event without treatment after withdrawal of phototherapy is low. On the other hand, for the gene therapy, they assumed that they would expect 50% probability of such successful withdrawal of phototherapy. So the idea of the study was that they would test two doses of the therapy, and then they would select the dose which seemed to be the safest, and then they would extend the cohort of the patients at this dose. And in fact, the idea was to inject the drug and then after 16 weeks withdraw, phototherapy will be stopped, and then the total bilirubin level will be monitored for at least a week. And the idea would be to, of course, introduce red start phototherapy if the bilirubin level would go over the threshold, and if not, the phototherapy would not be started. And in fact, the idea was to analyze the data based on the proportion of on the confidence interval for this proportion of successful withdrawal of phototherapy in this cohort of patients. So indeed, the natural history or knowledge of the disease indicated that phototherapy controls bilirubin level. Without it, the bilirubin level should go up. So if after gene therapy, withdrawal phototherapy would not lead to the increase in bilirubin level, this would be taken as the proof of success of the therapy. So to conclude, trials in rare diseases pose numerous challenges, right? And as we said, there are many diseases, they are very heterogeneous, so there's no one design fits all solution. Each case may, in fact, need some ingenuity to address the challenges. As Els mentioned, approval of any drug must be based on substantial evidence of the drug's effectiveness. However, when you read the regulations, as Els said, there is a flexibility to consider non-standard designs, and there is flexibility for innovation in these trials. And these examples, which I try to show, are testimony to that. That concludes my part, and that concludes also the entire webinar. So I think we can stop here. Alicia, I can try to give you or you can just claim the.
Okay, I think I've claimed, have I?
Yes.
Can you see my? Okay. So excellent. Thank you again to our speakers today for your detailed presentation. I hope everyone has enjoyed it. So let's begin the Q&A portion of this meeting. We have received one question, and I'm going to read it out. And the question is, with regulatory agencies offering accelerated pathways and flexible endpoints for rare diseases trials, what strategies do you recommend for sponsors to maintain scientific rigor while maximizing enrollment and ensuring timely access to therapies for patients with unmet needs? Do you want to comment, one of you, or both?
I guess Els can comment.
I can start at least. So I think this is a question where we can say that there's not one design or one approach that fits all. And I would say that for rare diseases, you can use all the regulatory options there are, like, yes, accelerated approval. If you have an early endpoint, if it's known in the rare disease, of course, that's the way to go, and you'll likely be successful. If you don't have these endpoints already defined, that might be an issue to start defining and validating two different endpoints, like the early one and then the clinical relevant one. But in terms of rigor, I think it's everything that Tomasz mentioned, right? The more complex you make the trial, the more innovative designs you try to implement, I think the more experienced the people have to be, the trialists around the trial, just to make sure that what you do makes sense, and really interact with the agencies as early as possible, just to see, you can make it a proposal, a decent proposal, and then see what they accept and where they can help and what they can potentially accept. But it's, I think, a very general answer to a very general question. I don't know, Tomasz, if you have other
No, you
Uh,
nailed it. You nailed it. And yeah, as we said, these diseases are heterogeneous, so it's really difficult to say. This is a strategy will help you. I would go back to what you said. I think again, people are used to, well, they are comfortable within the realm of randomized clinical trials. So if one can think about generating randomized evidence, I think that's already a big thing, because I think that's where many of worries about confounding and so on may be actually addressed. So that's something which is important, I think, in my opinion. Now, if you cannot do this, like as El said, you may want to go to some innovative solutions, but then you have to provide like more. You have to think about providing more data, more evidence. Collect additional data in order to support. Let's say as much as possible provide evidentiary let's say results, or provide evidence that which could convince the agency that what is observed is real and not a result of, for instance, a non-randomized comparison. So I think, yeah, it's again, as El said, it's difficult to give one strategy, but to me, I believe in randomization. So I think whenever it's possible, you should start from that.
Yeah, creative randomization is better than no randomization. Yeah, agreed.
We have actually gone slightly over the time, so I don't think we have any time for more questions. I wanted to thank everyone for your questions. We have reached the end of this question and answers portion of today's session. Please note that if we couldn't attend to your questions now, we'll be sure to follow up with you after this presentation. If you do have any further questions, do not hesitate to send them to info@iddi.com. So thank you everyone for participating in today's webinar. You will be receiving a follow-up email from us with access to the recorded archive for this event. A survey window will be popping up on your screen. Your participation is appreciated as it will help us to improve on future webinars. A final thank you goes to our speakers. Thank you very much for your insightful presentations.
Thank you very much. Bye-bye.
Thank you all. Bye.
So thank you now.